I'm pleased now to introduce Barney Graham, who's professor of medicine and microbiology, biochemistry, and immunology at the Morehouse School of Medicine in Atlanta. He's also director of the David Satcher Global Health Equity Institute at Morehouse. Barney was deputy director of the Vaccine Research Center at NIH, and his research was pivotal in the development of COVID and RSV vaccines. Thank you. Please welcome.
Well, good afternoon. It's a pleasure to be here. Thank you for everyone who stayed until the end of the day. Let's see. I am now at Morehouse School of Medicine, but I had 21 years at Vanderbilt where I started working on RSV with Peter Wright, who's here. I had another 21 years at NIH where I think almost stopped working on RSV with Jason McClellan. So it's fun to have both of them here today. I'm going to tell you about RSV, and maybe everyone already knows all these things because there's been so much advertising on TV about Arexvy and others, but I have these conflicts of being an inventor on vaccines and monoclonal antibodies and an advisor and consultant for those pharmaceutical companies. So RSV is a pneumovirus. It has about a 15 kilobase genome, 11 proteins that are known to be expressed. And I'm going to talk mostly today about the surface glycoproteins, particularly F, which you see here. So RSV causes disease in infants and adults, and it infects the ciliated epithelial cells in the small airways and the type 1 pneumocyte in the alveoli. These are cells that COVID also infects, SARS-CoV-2, but in the alveolus, the SARS-CoV-2 infects the type 2 pneumocyte, so it affects surfactant production. So it makes the pathophysiology a little bit different than RSV. You might also notice that RSV tends to not infect the basal cells in the respiratory tract, so it makes it a little easier to regenerate. We think most of the disease in children and maybe even adults is caused by the small airway obstruction from sloughed epithelium, inflammatory debris, fibrin, and mucus. Just to continue the comparison to coronavirus, you see coronavirus on the right. It's about an 80 nanometer in diameter sphere with those knobs on the surface, which is the spike protein. RSV, even though it's usually depicted as it looks in the lower left-hand corner, it's really a filamentous virus as it's budding from the cell. And as the matrix layer sort of degrades, it assumes this pleomorphic shape. It has two major subtypes that co-circulate every year and sort of change in the level of prominence in each year, in each setting. And unlike influenza or unlike what SARS-CoV-2 is doing right now, it's mostly toggling between these two subtypes. It doesn't drift. Every once in a while, there's an introduction of a new mutation in the F glycoprotein, but the two subtypes of the 495 amino acid ectodomain, there's only about 26 amino acid differences between those two subtypes, and it toggles back and forth. Everyone's infected by 2 or 3 years of age, and we are all reinfected throughout life, so most of the adults in here have been infected more than a dozen times throughout their life. That's what makes it so easy to boost with a single immunization of the proteins. Causes a lot of lower respiratory tract. It's the leading cause of hospitalization in children under 5 in the U.S. And in the U.S., there's not many deaths, but in the world, there's a lot of death. And most of it is undetected. It goes undetected because a lot of it in low-income countries happens at home. Severe disease in children, oh my goodness. Severe disease in children under 6 months of age is associated with childhood wheezing and asthma, and there is a legacy of vaccine-enhanced disease that I'll briefly mention. If you look at when RSV happens, it's a seasonal disease, at least in temperate climates, so I usually think of it between Thanksgiving and Easter. But sometimes, including the last few years, it has surprised us. You see the line for SARS-CoV-2 in the 2021 wintertime, where it never really emerged. And then, starting in 21-22, it came early, started up in September, and then in 22-23 winter, it came early with a big peak. And we still don't completely understand that. This year, it came up a little early in the southeast, but in other places, it's coming right on the usual schedule. It is a problem everywhere. These are data from the Perch study in Africa and Asia, where it shows that RSV is the leading cause of death by far among other respiratory pathogens. In adults, it's comparable to influenza, and there are a mortality of about 0.1%, hospitalizations of about 1 to 2%, and it causes a lot of medical encounters. And so comparing that to influenza, you see that on average, it probably has a little less disease than influenza, but in some years it's comparable. And a lot of the disease that we have attributed to influenza has actually been RSV, and some of it's been human metapneumovirus. So as we think about the periods of risk for severe disease, it's mostly in these young infants, especially between 1 and 6 months of age, but about half of the hospitalizations are occurring between 6 months and 5 years of age. And then at the end of, when we become more frail, we have another period of risk. So in the early 1960s, at a time when the Salk polio vaccine had just been so successful in nearly eliminating polio, people decided to make a whole inactivated virus vaccine that was made similar to the way Jonas Salk made that first whole inactivated polio vaccine. And in 4 child cohorts in 1965-66, immunizations were done. And the youngest age cohort, who began immunization at less than 6 months of age, of those 31 infants who were immunized, 20% of them were infected, and out of those infected, 80% were hospitalized, and there were 2 deaths. So this vaccine-enhanced disease really put a halt to most vaccine development over the next 20 years. And if you're interested, and I think you should be, you should go read this article in the link that was just published a month or 6 weeks ago. It was written by a journalist from MIT in an online journal called Undark. And he went back and uncovered this story about the vaccine-enhanced disease and was able to identify the 2 children who died, and was able to then identify their family members and talk to siblings of those children who died, who never really knew the story of what happened to their little brother. So it is going to be a story that probably continues to emerge and will probably be a challenge for the discussions around RSV vaccines. There's been two major immunological phenomenon associated with this vaccine-enhanced illness. One is an antibody problem, and the other is a T-cell problem. And the antibody problem was shown by Fernando Paulik and Ruth Karen in the early 2000s to probably be related to an antibody response that was not very effective at neutralization and resulted in immune complex deposition in the small airways and complement activation, probably adding to the damage from the inflammation itself. And so that's the antibody problem. And so it was known that you induced a lot more binding antibody than neutralizing activity during this infection event. And so one of the things we have to do with antibody induction is make it sure it has high neutralization activity. In animal models, it was shown that immunization in this pattern was inducing a primary Th2-like response, an IL-4 dominant response in the lung, whereas primary infection or reinfection is a Th1-like pattern of CD4 T-cell response, with interferon gamma dominating. And so this T-cell response, which could add to the mucus production and inflammation, because IL-4 and IL-13 can both inhibit cytolytic activity, so the inflammatory response is exaggerated because you're not clearing virus. It can create eosinophilia responses because of the IL-5 and a neutrophilic alveolitis, the mucus from IL-4 and the bronchoconstriction from IL-13. So this Th2 response can augment the disease. Finally, in the 1980s, some of the details of RSV started to be uncovered, the proteins and some sequencing studies, et cetera. Monoclonal antibodies were developed. And so by the 90s, a new set of vaccine studies were attempted in adults, not in the seronegative children. And they were all based pretty much on the F-glycoprotein, either purified from virus or made in baculovirus or CHO cells, mammalian cells. And these were all probably or proven to be in a post-fusion state, and they all had similar results. None of them met their objectives, and none of them were able to advance. And so that started changing in 2013. 2008, a young postdoc in Peter Kwong's lab at NIH got bumped down to the second floor next to my office, and we met, and Jason and I started a project picking apart the structures of the RSV F-glycoprotein. And over time, by the end of 2012, we were able to capture this pre-fusion structure of the F-glycoprotein bound by an antibody called D25 that is now the new nirsevimab monoclonal antibody is derivative of this. And Sean Deal, who's here in the audience, was involved in discovering that D25 molecule. And Tim Beaumont and Hergen Spitz at AIM Therapeutics and were involved in publishing that antibody. And then we found other antibodies either in the patent database or we found our own through a collaboration with this group at the Xiamen Medical University in China. And so by holding the top together with the antibody and holding the bottom together with the trimerization domain fold-on, we were able to find that structure and also found that these antibodies to this new site that hadn't been detected before were very potent, up to 100 to 1,000 times more potent than palivizumab, which was the licensed antibody for prophylaxis in premature infants. Jason showed some of this, but the prefusion F doesn't look anything like the postfusion F. And you can see why that happens. And he described this central helix and how this serves as the pivot point for all these domains to stack and create this heptad repeat that goes up and is responsible for projecting the hydrophobic fusion peptide into the host cell membrane. So the molecules are very different. And having that structure and also having the postfusion structure has allowed us to map epitopes. We now know pretty much the whole antigenic surface of the RSV F protein. This site we call site 0 and site 5 are the most potent targets for neutralizing antibody. This little section down here, the stem, is relatively newly defined with some new antibodies that were just published. And some of these sites only exist on the pre-F molecule. Some only exist on the post-F molecule, this 6-helix bundle region down here. And the hierarchy of potency is that the pre-F exclusive binding antibodies are more potent for neutralizing activity. It also explains some of the observations that had been made recently. And looking at cryo-EM, you could see that there was a short and a long form of the F protein on the viral membrane depicted here, and those pretty much perfectly fit with the size and shape of the prefusion and postfusion molecules. So the F protein is flipping on the viral membrane surface spontaneously. It's very metastable. And as I mentioned, it's a filamentous virus. You can see the filaments budding from cells. You can see them in scanning EM, bridging across to a new uninfected cell. You can see how the fixation process actually broke some of those filaments that were trying to bridge from one cell to another. You see the density of F in this transmission EM, immuno-TM. And then you see this cryo-electron tomography work that can show you that most of the pre-F is populating the filament is the short version or the prefusion molecule, but a lot of the F on the polymorphic or rounded up virus is now in the post-fusion state. So this is just an illustration of that concept that when the matrix layer that's underneath the membrane starts breaking apart, it causes the F to flip from this pre to post form. So what can we presume about the vaccine-enhanced illness knowing about these new structures? This is the way the vaccine was made back in the 1960s. And the major, well, that box kind of jumped, but the major feature was that it was cooked at 36 degrees for 72 hours and with a very low dilution of formalin. That red box is supposed to be one down. And what you can see is that when you incubate RSV at that temperature, what you can see is that the virus dies. It can no longer replicate. After 24 to 48 hours, it's almost completely replication incompetent. And so the question is, what happens to the F protein during that time? And this explains what we think happens, because when you use an antibody that can bind both the pre and the post-fusion form, a dual binding antibody like modivizumab, which was derived from palivizumab, you see F is present all the way through. But with pre-F exclusive antibodies like these three, you can see that the kinetics of F flipping from pre to post is the same as that of the virus dying. So we think that the pollen-activated virus vaccine that was used in the 1960s was essentially a post-fusion F antigen. So now we have this new structure and have identified a new site of vulnerability at the apex. And we know that when that molecule flips into the post-fusion form, the epitope is lost. And we know that using that molecule in repeated efficacy trials has not worked. But if you can stabilize the protein with internal disulfide and cavity-filling mutations in a trimerization domain at the C-terminus, you can now achieve an antigen that can induce neutralizing antibody responses 16-fold or greater in most adults. And Jason showed you something like this earlier, but I want to just repeat it because it's such a remarkable phenomenon, and it took multiple rounds, even among our RSV colleagues, for them to see why this was happening and what a difference it made. So if you've got the virus on the bottom and the cell membrane on the top, this top half of the molecule unravels. The hydrophobic fusion peptide goes into the host cell, and the heptad repeats now pull the membranes together to get a fusion pore, and the nucleocapsid of the virus can now move from the virus into the host cell and start the replication process. So it's really common sense that you want to start and attack the native protein before it's turned into this post-fusion form that is non-functional and spent. And so it makes sense that the pre-fusion would be a better target than the post-fusion, but you can also see that the bottom half of this molecule doesn't change that much. So there are some neutralizing epitopes left on the post-fusion that also exist on the pre-fusion, and that's why the development process was so confusing to those who didn't have structures to work with. I don't have time to show all this, but because of the new technologies we have for single-cell B-cell analysis and the new probes we now have with pre- and post-fusion molecules that can look at the B-cell repertoire in response to the vaccine, you can go to some of these papers and see how these subunit proteins that were boosting prior immune responses can behave. And these reagents that come from this work, as Jason said before, a lot of the same reagents that were used to make the COVID vaccines were also used as bait or probes to identify human monoclonal antibodies that were then turned into immunotherapy. And you can use these probes. So for instance, this is the pre-fusion on this axis and the post-fusion on this axis, and a yeast display system that pretty much creates these fingers. And so this upper left quadrant would be pre-fusion exclusive, and this would be post-fusion exclusive, and the middle ones would be dual binding antibodies. And you can separate these out by having these other monoclonal antibodies characterize these clouds of, in this case, yeast, but you can see the same thing with B cells. And you can actually now map then epitope-specific B cell responses and characterize those responses by phenotype or in any other way. So it really does create remarkable tools when you have the structure-defined probes in the single-cell analysis. So when you look at one of the last post-fusion F vaccine studies, MedImmune was kind enough to share some PBMCs with us. If you look at pre-fusion immunization, you achieve this pre-fusion exclusive B-cell cloud, and with post-fusion you don't get that, but you do get a lot of dual binding B cells to either site 4 or site 2. And if you can achieve these pre-fusion exclusive, looking at 800 or 900 monoclonal antibodies that we isolated with Laura Walker's group from Dartmouth when Jason was here, you can show that pre-fusion exclusive binding monoclonals are on average 10 to 100-fold more potent than antibodies that bind both the pre and the post-fusion molecules. So it explains why this vaccine is working better to induce better antibodies. And there's many ways. We think this way worked pretty well with the disulfide and cavity filling mutations, but other groups have used slightly different internal stabilizing mutations. And as Jason mentioned, Hans Longedik at Janssen, formerly at Crucell, even had a proline substitution at this top of the central helix that stabilized the molecule. And so over these last 10 years, a lot of work has gone in from several different companies using this new information to develop products. And just this year, we now have 4 FDA-approved products. Some of them are also approved in the EMA, either for vaccinating the elderly. GSK has a subtype AF protein with their AS01E adjuvant. Pfizer has subtype A and subtype B subunit proteins without an adjuvant. And then AstraZeneca has a monoclonal antibody, nirsevimab, that was derived from D25, all of which now are available, even though some of them are in short supply. So the elderly are being vaccinated. I haven't heard much about rates of maternal vaccination this year, and so far nirsevimab has only become available for very premature, high-risk infants, even though it's approved for full-term infants. And I got to be vaccinated a few weeks ago, which I was really happy about. So, and you can see that the lower respiratory tract disease, especially the severe lower respiratory tract disease, these protein-based vaccines have achieved a pretty high level of efficacy. And some of that efficacy is lasting into the second year now. And so we have maternal vaccination that extends the protection of the 0 to 6 month old infant. We have passive antibody that can be given directly to the infant that extends and reduces that risk period. And we have elderly vaccines for the frail elderly in particular that can take care of that risk period, but we still don't really have a vaccine solution for the 6 month to 5 year old infant, where still a lot of hospitalization can occur. So in these different vaccine target populations, we have vaccines, but this group here is still a work in progress, because doing the priming immunization has a higher risk because of what happened back in the 60s, and also higher stakes. And so these vaccines are likely to be the priming event for the vaccine is going to be probably based on gene delivery, either with attenuated virus or chimeric virus, RNA, or viral vectors. So in conclusion, this is a great story for the role of basic science in applied practical use of products, and it can be used to convince people that increased support for basic research is critical to everything we're doing in the future. We now have this proof of concept that was for structure-based vaccine design. We have a variety of products now for preventing RSV, either in the elderly or in maternal or in babies. And this process, as Jason said, when he came to Dartmouth at the end of 2013, we had just done this work on RSV, and we're looking to extend it into another envelope virus when MERS came along and convinced us coronavirus is the right thing to work on. And so it was really the basis for a lot of the rapid development of the COVID vaccines. And there's a lot more to do. So for all the young people in the room, which means anybody younger than me, there is a lot more to do. And the scientific tools we have to do it are just extraordinary, where you can define your antigen at an atomic level, where you can define your B cell response on a single cell level and define repertoires and really define all the immunology and biology you need to understand boost interval and all these other things that have been a mystery for so long. But it takes time. So if you want to make a vaccine, you have to give yourself at least 30 years because it doesn't always happen in a year or even 10 years. You've got to give yourself some time. And let's see. This is a whole nother talk, but at some point we have to shorten the gap between vaccines being available in high-income countries and low-income countries, because there's only about 4 million babies born in the U.S. every year, but there's 120 million babies born in the world. And, ignoring the majority of infants for the sake of the minority of infants is just not sustainable. So I want to thank our collaborators. All the work I've done on structure-based vaccine design over the last 15 years has been with Jason. And this group at the VRC and this group of investigators, very talented investigators who helped identify some new antibodies, really were involved in that early work to get the first structure. This group, Michelle Crank is the PI of the clinical trial, and Tracy Ruckworth leading this group on analysis of the clinical trial, were really responsible for most of the data that I showed you. I had the privilege of working with this very talented group of investigators at the VRC and a very wonderful group of students and postdocs in my laboratory. And I just also want to mention that almost everything I know about vaccines started at Vanderbilt. And I want to mention my new colleagues at the Morehouse School of Medicine, where we're mostly going to be working on how to change equity, vaccine equity, and then all the people and investigators involved in the vaccine evaluation. So I'll stop there and thank you.